Okay, so in prokaryotic cells, the gene control relatively simple. Gene control relatively simple, as we do know that, okay, in prokaryotic cells, okay, they involve promoter, operator, and then structural gene. So as long as operator has nothing, no repressor protein, then transcription can take place because RNA polymerase can bind. Okay, so to control the entire structural genes, then we depends on this switch, the operator. So relatively simple in the process. Okay, but in eukaryotic cells, then slightly different, because in eukaryotic cells they may not have the operator. So if no operator, how we can control? Okay, how we can control? And sometimes we want to control the structural gene may not be together. So we want to control entire thing, and then we want to in a sequence. Then eukaryotic cells, you have more cells, more genes in the cells. So therefore, you can see that, okay, we need to have a very tightly regulated, okay, the entire things must be tightly regulated. Okay, so to move on from here, then we start to look at this, okay, gene control in the eukaryotic cells and how it happened. So scientists have done a special the research on these eukaryotes. Okay, so what they have done? Okay, they culture the animals, the eukaryotic cells. When they culture eukaryotic cells, then they put in the RNA polymerase and check and see whether, okay, whether any gene product or not. When I say gene products, basically means that those protein, you see, and then they realize that, oops, no gene products is formed. Okay, no gene products is formed. Okay, so why? If you do the same thing on the bacteria or prokaryotic cell with an RNA polymerase, then okay, but here cannot. Okay, because eukaryotic cells are more complex. We do know that they are more complex, so means that they must be tightly regulated. So first, all genes are transcribed by single RNA polymerase in bacterial. Okay, but eukaryotic cell can see that they have multiple or different kind of RNA polymerase. That's one. Second, you can see that the RNA polymerase, instead of binding, rather than binding directly to the promoter, okay, instead of binding directly to the promoter, they realize that they need to have an additional protein here. Okay, so initially they call this additional protein as initiation factor, means that to initiate, okay, to initiate these transcriptions in eukaryotic cell, then they must have this initiation factor, which this initiation factor now is commonly known as the transcription factor. Okay? It's commonly known as transcription factor. They are required for RNA polymerase to initiate the transcription. So what is the transcription factor? Basically, they are protein. They work together with the RNA polymerase so that we can initiate the transcription process take place. Okay, so a transcription factor, basically they are proteins, highlights, they're proteins. They control the rate of the transcription. So it means that this transcription factor can increase the expression, it can decrease the expression. So their function is to regulate a particular gene, in order to make sure they're expressed in the right cells at the right times. Okay, so I always put it in this analogy. Okay, you try to imagine that in bacterial cells, they do not need to have the tightly regulation or tight regulations. So it means that RNA polymerase can say what they can do whatever they like to initiate the transcription. But in eukaryotic cell, then it must be both RNA polymerase together with the transcription factor I call it as a TF, right? Transcription factor, TF. It's something like you try to imagine your bank account. Your bank account is not tightly regulated. Only you have the key to open, this safety vault, for example. So if you alone have the safety word key, so whenever you want to open, you can open. So it means that no tightly regulation. But now try to imagine the key you have one, your father have one, or your girlfriend have one, for example. So it means that in order to take anything, you must have both keys together to open the vault. So it means that the vault or these safety cabinets will be more tightly or more safely regulated. Okay, so same goes to eukaryotic cells, because we cannot simply activate on a certain gene or off a certain gene. So therefore, you can see that we need to have this, what we call the tight regulation. So how you can see that we need to have this transcription factor. Sometimes more than one transcription factor is needed. Okay, so transcription factor work alone or with other protein to in a form of complex. They can promote the gene expressions. It can block the gene expression. So it can be a repressor, it can be an activator, it can be a repressor protein, it can be activator protein, but they do not bind to. Always remember, different from the prokaryotic cells, they do not bind to. Okay, they do not bind to operator. But where they bind? They bind to the promoter. Okay, they bind to the promoter. So what are the involvement of transcription factor? For example, they involve in the transcription process. As usual, the general role. They make sure that they activate appropriate gene in a sequence. So for example, who comes first? Okay, you must have this gene come first, this protein come first, and then followed by another one. For example, in terms of the cell cycle, okay, we have to proceed from the interphase, then go to the prophase, prophase go to the metaphase. So it means that they must control the cell cycle in a correct order. So transcription factor can do this for us. Also, transcription factor is responsible for determinations of the gender in the mammal. Okay, so all mammal, we do start it for male, we do start it as a female first. So it means that the female chromosome start, the X chromosome start to be activated first. But along the durations, the fetus developments, in the fetal development, you can see that the Y chromosome start to take the effect because of the transcription factor. Okay, so transcription factor also allow the response to the environmental stimuli. So we're going to do this or use this as example study later. Okay, how we respond to the environmental stimuli, and then we can switch on and switch off the correct gene. For example, we do know that certain gene like melanin in our body, when exposed to the sunlight, UV, okay, then you can see that there is an expressions of the gene, the peroxidase, for example, and then allow us to have a more melanins. Okay, so some transcription factor, including the products or the proto-oncogene tumor suppressor gene in this case. Okay, proto-oncogene means cause cancer. This one actually suppress the development of cancer. Okay, so this must be in the correct ratio. Okay, and also you can see that a lot of hormones, they also actually have the effect through the transcription factor as well. Some hormone actually bind the transcription factor and then allow the gene activations. Okay, so we're going to look at these hormones in the next few chapters, where we're going to look at the hormonal control. Okay, look at hormonal control in that particular chapter. Okay, so how? Now we're going to look at this as one of the example. How this DELA protein actually, they are repressor protein. They are transcription factor, but they are repressor. Repressor means what? When I say repressor, basically means that they block transcription. Okay, they block transcription. So for example, this DELA protein is very, very important when the seed dormancy take place. What does mean seed dormancy? Seed dormancy means that they have to reduce the activity, protect the seed, don't want to get the seed to be germinated. So for seed to maintain at this dormancy, then we have to block the transcription. We need the DELA protein. Okay? We need the DELA proteins. Okay, but to break this seed dormancy, then we need the hormones called gibberellin. Okay, gibberellin. So I'll call it a GA. Let's look at what actually happened here. Look, okay. So DELA protein and seed germinations at A basically absent of gibberellin. So A means that seed dormancy. Okay, when under seed dormancy here, sorry, and under seed dormancy, you can see that we do have the DELA protein. Look at DELA protein. So DELA protein bind to this transcription factor named as PIF. Okay, phytochrome interacting factor. Okay, phytochrome interacting factor is a transcription factor. So what will happen here is when DELA protein bind to the transcription factor or this PIF, so PIF cannot bind to the promoter. Here is a promoter. So if a PIF cannot bind to the promoter, therefore RNA polymerase cannot bind. Therefore, you can see that there is no gene activation. So if no gene activations, no transcription, so basically means that no germinations. Okay, so at this moment, I think I need to actually tell you guys something before we proceed here. Okay, so let us look at this. Now, in eukaryotic cells, okay, so you can see that this is a promoter, and then you have particular gene. So for this gene to be activated, for eukaryotic cells, slightly different, because in terms of promoter, RNA polymerase cannot bind directly. Okay, so they cannot bind directly. Okay, they cannot bind directly. So in order for gene transcription to take place, we need to have the transcription factor first. So means first step, transcription factor have to bind. Okay, bind to promoter. And then step number 2, RNA polymerase now can bind. Can you see that? Are you clear? So means that these RNA polymerase, they have no way to bind directly to the promoter. So when this RNA polymerase cannot bind directly to promoter, therefore no gene expression take place. So but now with the transcription factor, with the RNA polymerase, then yes, we're going to form mRNA, we're going to get the polypeptide, and get the protein, for example, to make sure that cellular activity can take place. Okay, yeah. So now we'll look at the seed dormancy. When during the seed dormancy, so means that the seed want to make sure that their metabolic activity is at very, very low. Okay. Must be very low. Okay, when the seed dormancy, metabolic activities must be low. So means that they do not require enzyme. No enzyme, proteins needed. Okay, under seed dormancy. So how we can make sure that there's no transcription? How? So the seeds have the mechanism. In this case, they have the protein called DELA protein. So what's the function of DELA protein? DELA protein actually is a repressor protein, or we call it DELA repressor. So what DELA protein actually did, DELA protein bind to a transcription factor. This transcription factor called PIF, okay? Phytochrome interacting factor. Okay, so what is PIF? PIF actually is transcription factor. So you can see that now transcription factor bound to the DELA protein, transcription factor now cannot bind to the promoter. So it means that in this case, PIF want to bind? Sorry, cannot. Why? Because the transcription factor, the PIF now bind to the DELA protein. So if now the transcription factor cannot bind to the promoter, means that my RNA polymerase, even though the RNA polymerase is still there, useless. Why? Because RNA polymerase also cannot bind. Are you clear? Okay, without PIF, RNA polymerase useless. So even though have RNA polymerase, no gene transcriptions, so means that no mRNA now, therefore no enzyme. So metabolic activity remain low in the seed because now seed under dormancy, which the seed doesn't want to be germinated. Okay, so to break this dormancy, to activate the dormancy, so the opposite way here actually is the seed germination. So seed germination, I won't cover in a detail, okay? I will cover until the expression only, okay? So later stage, when we come to the final chapter, then we look at the gibberellin, okay? So if you look at the seed, look at the structure of the seed. So the seed will have the embryo, and then we have a layer of seed coat. So this is embryo. So in the seed, embryo will grow when germinations will grow into the adult plant. Germinate lah, okay? Okay, after germination. But for the seed to make sure that germinate at the correct environment, okay? In order for the seed to germinate at the correct environment, the first thing, they must have sufficient water. So the absorption of the water then makes this embryo actually produce a hormone called gibberellic acid or gibberellin. Can you see that? Water molecule absorbed in the embryo. So embryo now know that, okay, I have sufficient water supply already. I can germinate already. So how to break the seed dormancy? First thing, okay, water absorbed. Second, you can see that they produce the gibberellic acid. So this gibberellic acid, what is the function of gibberellic acid? So gibberellic acid basically in this case involved in the seed germination. How they involve in the seed germinations? So first thing, okay. So in this aileron layer of the plants, okay, or the seed, sorry, or the seed, they have the GA receptor. So you can see that. Let me draw it first. So what is this? This is a gibberellic acid receptor, GA receptor. So what happened to this GA? When gibberellic acid bind to the receptor, again, okay, when it bind to the receptor, it's going to trigger a series of events. Okay, the complex here, we call it as complex. So this complex now will trigger a series of events, or series of reactions. So what's the function of this series of reaction? They trigger a series of reactions so that they can break down DELA protein. Can you see that? Okay, so why this complex? What's this complex called? What's the name of complex? This complex is the gibberellic acid or gibberellin receptor complex. So when this complex, okay, is formed, then it will trigger a series of reactions, result in the breakdowns of the DELA protein. So it will break down DELA protein. So initially here is the DELA protein bind to the PIF, correct or not? Just now. But now we break down the DELA protein already. So means that now PIF is free. So PIF now can bind to the promoter. This is a gene, structural gene. So when PIF can bind to the promoter, therefore RNA polymerase can bind. So it means that PIF cannot do anything also. You can bind the promoter, but without RNA polymerase, useless. With RNA polymerase, without PIF, also useless. So both of them have to work together and then allow the transcription to take place. and then allow us to have the enzyme that involved in the seed germination. Are you clear? Can I see the mRNA will be translated, the polypeptide fall into the enzyme and allows the seed germination to take place. Okay, so now you can, if you need to take a photo, you can, okay, to compare between these two processes here. Okay, one is seed dormancy, another one is seed germinations. So based on what? Based on this Della repressor protein. Okay, so again, I explain it again here. The Della repressor proteins, what they do, right? First, they bind to the transcription factor known as PIF. Okay, phytochrome interacting factor. So it means that when a PIF bound to the Della proteins, they are not able, right? They are unable to bind to the promoter. Therefore, RNA polymerase won't be able to bind. So therefore, you can see that RNA polymerase cannot bind. Then no mRNA and no enzyme. So seed continue to be dormant. Okay. But when the surrounding environment, can I say that, stimuli? So the stimuli here is the water. So sufficient water absorbed by the embryo, therefore the embryo release the hormones called gibberellic acid. So this gibberellic acid now involved in the seed germination. How? They bind to the receptor. When they bind to the receptor, they're going to trigger a series of reactions, result in the breakdowns of the Della protein. So when the Della protein is broken down already, then PIF is free. So PIF now can bind to the promoter, followed by the RNA polymerase, allow us to have mRNA and the formations of the enzyme that involved in the seed germinations. Okay. So you can see that this is more complex. Just now I already summarized for you guys already. So this is a complex process. You can see that PIF bound to the Della protein. PIF cannot bind to the promoter, therefore seed dormancy continue. But when this gibberellic acid, can you see that gibberellic acid with the receptor, GID1 is a receptor. Okay, this one you don't need to remember GID1 is a receptor. So when gibberellin or GA bind the receptor, it will cause the breakdown of the Della protein. So free what? Free the transcription factor. So transcription factor can bind to the promoter, allow the growth activators to take place here, basically involved in the enzyme, in the hormones or proteins that involve in the seed, okay, germinations. Okay, so I go through the answer with you guys, can check later. I don't want to go to the detail. We'll come back to this one again when we go to the chapter 19, okay? The final chapter of A2, we're going to look at how this gibberellic acid involved in the more detailed way for the seed germinations to take place. Okay, so with this, I have done for the first part of our lessons now. For today, okay, stop recording now.